Assalamualaikum. Now let's continue meiosis as we started meiosis in the previous video only. So now we'll continue with it as we know that meiosis is a reductional division. But meiosis has two phases. First phase is the reduction division and another phase is the equational division. So in first phase the number of chromosomes become haploid. Means the number of chromosomes diploid means suppose in human being 46 chromosomes are there. So that is diploid. And in first meiotic division that become haploid become an only means suppose this cell having four chromosomes, so out of four, two will be moving towards one pole and two will be moving towards one pole. So means like the spindle fibers are attached to the one from the one side only to the centromere. So they just half of the chromosome move towards the one pole and another half set will move to the another pole. In this way, the number of chromosomes become half like this. Means out of four, the two move towards the one pole and another two will move to the different pole. So in this way, half of they just divide into half and they become haploid. And the number of chromosomes become half. So meiosis is categorized into two phases: meiosis first and meiosis second. Meiosis first is having the four phases. So meiosis first having the four stages like mitosis, prophase first, metaphase first, anaphase first, telophase first. And meiosis second, prophase second, metaphase second, anaphase second, and telophase second. So here prophase first further categorized into five categories, that is lactotine, zygotine, pactine, diplotin, and diakinesis. So these are the different phases of the prophase first. So let's understand what these all stages are, what happened in these stages. So let's start with the prophase first. Prophase first that categorized into five different categories. So here in this the chromosomes start appearing. Because we know that the DNA material in the form of chromatin network, and at the time of classification cell division, sorry, they just start condensing to form chromosomes. So at this stage they start appearing, and then the nuclear membrane start disappearing, and this centrosome, in the centrosome centrioles are there, and they just split to move towards the opposite pole. So this all happen in this phase. But along with that, what happen here, they just on the homologous chromosome they come or they attract each other to form pair. So means one of the set is come from the male and one from the female. So they just attract and they lie near to each other. So in lactotine, the chromosomes gradually visible, and after that these chromosomes make pairing, means pairing of homologous chromosomes occur in after that. So that stage is zygotine. The first is lactotine, means when the chromosome visible, then it is called, then that stage is called lactotine, and when the pairing of the homologous chromosomes start, then it is known as zygotine. Before moving to the next stage, let's understand what are homologous chromosomes. Like here in this picture, you just see that two chromosomes are there. So what they are actually, one from the father and one from the mother, and they are not identical. Of course, the father and mother two different personalities, and they have the what you can say, the different gene sequence in it, but they are similar in length and gene position. Means the DNA fragments, they that is also same, and their length is also same. So they are identical or similar in length, means they are, the length is same, their gene positions are same, and they are having the same centromere. Like you can see here, their length is same. These are the gene, the fragment of the DNA. They are same position, and their centromeres are also at the same position. So they are similar in this context. Otherwise, they are different from each other. because they come from two different organism, you can say, but they are of same species, but they are two personality with the different set of characters, so they are not identical exactly. Now let's discuss the next stage of the prophase, that is packaging. Here in this stage, that crossing over of non-sister chromatids is started. Non-sister chromatids means here you can see the two chromosomes. So the one which is having the two, these are the sister chromatids, means which are attached with the point centromere. So they are of the part of the same chromosome, so they are the two sister chromatids. But here they are the two different chromosomes, so they are non-sister chromatids. Means this red and the black, they are actually the different chromosome. So they are non-sister chromatids, and the crossing over start in the non-sister chromatids only, and this stage is known as placental. And after that, this chiasmata formation occurs. This crossover structure started forming. So at this diplotin stage, this chiasmata or crossover structures are formed. This cross structure is formed. And what happened at this stage? These crossover part, they just detach from it and then they attach to the different chromosome. Means like this red chromosome and then this is the black chromosome. So one from the parent, one from the mother, one from the father. So the part of the chromosome from the mother, it just detach the cross one part and attach to the another set. And in the same way, the part of this chromosome is detached and attached to the another chromosome in this way. So now they having the variation of the gene. Means here the gene, the characters will be changed. As we just noticed that the different sibling of a same parent having the different characteristic. So why they have the different characteristic? Because of this chiasmata formation. So because now because of this the variation in the characters are visible in within the species. So this is the last stage dikinesis of the prophase first. Here the terminalization of the chiasmata occur. So this all happen in the prophase first stage. And then after this prophase first, metaphase first stage is there. In this homologous chromosome means after chiasmata formation with this variable with the variable characteristic or with the variable um, uh, gene sequence, they just what you can say arrange on the equator in this way. The pair of chromosome, they one lie above the other and in this way, so that the spindle fiber are attached to the one chromosome, the centromere of the one chromosome from the one side, not from the other side. Means they are just attached from the one spindle fiber from the one pole, and half of the chromosome they are attached to the another pole. And then, like suppose in this cell four chromosomes are visible, so out of four, two will be moving towards the one pole and two will be moving towards the another pole. So in this way reduction occurs, means the chromosome become half. Like here is in anaphase, you just see that they just started moving towards another pole, and telophase they just reach to the opposite poles. After that, second meiotic division starts, and this is called first meiotic division, where the reduction occur in the chromosome number, where the number of chromosome become haploid. Means when you see here, this is the cell which is started meiosis. Here the four chromosomes are there, and after this meiosis first, the cell is having two chromosome in each. So the number of chromosome become haploid in meiosis first. After that, the meiosis second is start, means the second meiotic division is start, and that exactly like mitosis. Means as we discuss the different phases of mitosis, in the same way these stages will going on in the meiosis second division. So means second meiotic division will be same exactly like mitotic division. So here in this picture, it's not shown that they are the complete cytokinesis they have not shown, but after this the complete cytokinesis occur, and as a result two haploid cells are formed. So after first meiotic division, two haploid cells are formed, and then each haploid cell further go further. This second meiotic division is started in that. So here each cell again will go to the further division. What is that? Meiotic division, and that exactly like mitosis. Means the number of means the chromosome as they just started moving towards the equator, and then like these are the chromosome, they started moving towards the equator, and then they arrange on the equator, and the spindle fibers they attach to the centromere from both the side, and in this way the centromere is split. Now in second meiotic division the centromere is split. The centromere is split, and half of the chromosome will move towards the one pole, and half of the chromosome will move towards another pole, like here. They arrange on the equator, and then they just the spindle fiber are attached to the centromere, and then the centromere is split, and then the sister chromatin they start moving towards the opposite poles. So they reach to the opposite pole, and this we know that when they arrange on the equator, that is the metaphase, and when they started moving, then this movement started in anaphase, and in telophase they completely reach to the poles only, and then the cytokinesis occur, and as a result, the two more cell arises. Means in meiotic first division, the two haploid cells are formed, and now each haploid cell further go in the one more division, and then they will be converted into four. Means one become again, means this the cell further go in division and will give the another exact, means another cell, means the two same number of chromosomes would be there.